Hello, and welcome to the intolerable world of biology, aka the subject you wish you didn't have to take. Please. Anyway, today we'll be talking about biomolecules. So, there are a total of 10 main biomolecules you need to know, including poly, mono, and disaccharides, alpha and beta glucose, and ribose. But there are only 5 that you need to know how to draw, which are alpha and beta glucose, ribose, a general amino acid, and a general fatty acid. Firstly, we'll be diving into glucose, and the 2 kinds there are, which are obviously alpha and beta. So let's talk about the most confusing part of drawing these molecules, how to structure the hydroxide groups. In alpha glucose, you structure them in the down, up, down, down structure. You can remember it as alpha begins with the first letter of the Greek alphabet, so therefore there is only one up in that section. Or you can remember dud as in D-U-D-D, or doo-doo, which is D-U-D-U. So, therefore, beta glucose's hydroxide groups are drawn down, up, down, up. Now, alpha and beta glucose are used to make different polysaccharides. For example, alpha glucose is used in glycogen, whereas beta glucose comprises the molecule cellulose. Moving on, ribose. It is exactly like glucose, but with like 1 less carbon. It looks kind of like this. As you can see, it's a pentagonal structure, aka a pentose sugar. So think of how a rectangle like ribose has 4 sides, and then you add 1 to get the pentagonal shape. Now, ribose can be seen in tRNA, RNA, mRNA, rRNA, and all the RNAs. So, a general amino acid. There is a section in which the amino acid has an R group, which is where one of 20 different groups can attach. The general amino acid can be drawn by using a nitrogen molecule with 2 hydrogens coming off the side, and then 2 carbon atoms. So the total molecule number should be 2 carbon molecules, 4 hydrogen, and 2 oxygens. Now we have general fatty acids. These are used for energy storage in the organism. These are long chains of carbon with OH and double bond O on one side of the fatty acid. Now there are 2 types of fatty acids, saturated and unsaturated. Saturated fatty acids have single bonds, and of course unsaturated will have double bonds. You learned that in GCSE biology. This affects the shape of the acid, as unsaturated ones are able to bend in different shapes. These are all ones that you need to be able to draw, but now we'll just be talking about carbohydrates, and we'll make a separate video on water because it's such an interesting molecule. The carbohydrates are also crucial to our survival. You don't need to know how to draw them, as I mentioned, you just need to know what they are. There are 3 different kinds: poly, mono, and disaccharides. The mono and disaccharides are used for short-term stores of energy. An example of a monosaccharide is, let's say, alpha glucose, and disaccharides is maltose. Finally, there are polysaccharides, which are actually longer-term stores of energy. And a key example of this is glycogen, which has a similar shape to amylopectin, just with a couple more branches coming off the side. And now over to Deuce with our special guest teacher, Mr. Lang.
Welcome to the next segment, where we have our resident biology teacher answer some questions. Join me in welcoming Mr. Lang to answer the following questions about biological molecules.
Right, so there's 3 polysaccharides you need to know details of, right? Starch, glycogen, and cellulose. Starch, 2 forms of polysaccharide within starch, so amylose and amylopectin. Amylopectin is slightly branched. Always branching within a polysaccharide is going to create density. So starch has some branching. Glycogen, on the other hand, the storage molecule in animals, fungi, and bacteria, is highly branched. So you've got lots more branching in glycogen than you do in starch. That means it's a more dense storage molecule. Cellulose is slightly off to one side as a structural molecule. So cellulose is mainly in plant cell walls is where we'd find it. It forms part of what's called dietary fiber, so undigestible in most animals. Right, glycosidic bonds. Glycosidic bonds are made between 2 adjacent sugar molecules. So we need 2 hydroxyl groups, 1 from each sugar molecule, and then we're going to get water off as a product. So those 2 hydroxyl groups usually are made between carbons 1 and carbon 4 of the 2 adjacent sugars. Those hydroxyl groups then will come together. We make the glycosidic bond from the oxygen, and then we get water off as a product. We can also, whenever we get that branching structure, form what we call a 1,6 glycosidic bond, which is where we get a hydroxyl group from carbon 1 and then 1 from carbon 6, which will then create that branching structure. Right, 3 polysaccharides. Cellulose, only in plant cell walls. Glycogen, storage molecule of carbohydrates. We find that in animals, fungi, and bacteria. And then starch is the equivalent of glycogen in plants. So starch is the storage molecule within plants. Okay, so all of metabolism is grouped into these 2 types of reactions. Anabolism is building small things up into big things. So think things like protein synthesis, taking amino acids, building them up into a protein. Catabolism is breaking stuff down, so the opposite of that. So think digestion, anything where we take a big molecule and we break it down into multiple small molecules. Okay, all amino acids are made up of 3 component parts largely. So we've got the amine group, that's bonded to a central carbon, which we refer to as the alpha carbon. Off that alpha carbon, we have an R group, that's a variable group, so that's the one that changes depending on which amino acid you have. And then on the other end, you've got your acid group, your carboxyl group. So that's with the C, the double bond to the O, and then the hydroxyl group coming off there to create the acid end.
Now we're moving on to the question part of this lovely video we prepared for you. Today the question is going to be, outline the production of a dipeptide via condensation reaction showing a general dipeptide. Okay, so now that you've paused, tried to answer the question, or still don't know what's going on, I'm going to run you through the mark scheme and essentially how to get there, and then after this there will be a link to further questions. The first marking point is defining condensation. So condensation is the joining of 2 amino acids in order to form a dipeptide. Now you'll get a mark for that, and that's not actually a part of the question, but it's a definition which allows you to have a marking point, and that's key for the IB. So the second marking point is that the carboxyl group of 1 amino acids reacts with the amine group of another in order to form this dipeptide bond. Now, for this marking point, you will need a diagram of 2 generalized amino acids in order to show the condensation reaction between the 2, as you can see in this animation right here. Now, of course, as you know, in condensation, the water is eliminated, and that brings us to our third marking point. Water / H2O, depending on how you want to write it, if you're a chemist, biologist, or just don't know what the language is. Water is eliminated, and that is a simple 3 words that you need to get 1 mark. That brings me to our fourth marking point. Now, if you got already 3 of these marks, you got 3 out of 5. The fourth marking point is that you have to have a diagram of a dipeptide, and it has to be correctly shown, because obviously you're not going to get marked if it's incorrect, and that will be shown behind me somewhere. That brings me to our 5th marking point, and don't worry, I've only got 1 more after this. The 5th marking point is that peptide or covalent bond is produced, so the peptide bond has to be correctly labeled as well. Finally, the last and final marking point, if you haven't already got all 5, you geniuses, is that it occurs in the ribosomes, or at the ribosomes according to the IB. So, now that I've run you through all of this beautiful question, take your time and go through the other questions as well. They're in the link below. Please click to subscribe and like the video.
You've been watching LabM8s.